on because okay, we have the record button on, and so many of you know Caitlin. She's been affiliated with WBA for quite some time, but I'd like to introduce her for those of you who don't. She is a Pacific Northwest native with an early fascination for insects, which developed into an interest in studying the habitats and life histories of butterflies and moths. She is a GIS analyst by training and a conservationist by nature, and she enjoys photographing and collecting butterflies and working on various butterfly projects. She is a member of the Lepidopterist Society and lives in Southwest Washington. She has 2 self-published books. One is The Pocket Guide to Butterflies of Washington, which is a very handy reference that I usually carry around with me out in the field. And second book, Butterflies of the Similhekan Wildlife Area. And then she has co-authored with Bob Pyle, Butterflies of the Pacific Northwest. Caitlin earned a Bachelor of Arts degree in Geography from Central Washington University and a Master's of Science in Environmental Science from WSU Vancouver. The title of her presentation tonight is Looking for Lucia in All the Right Places. Thank you so much, Caitlin, for being with us tonight.
Thanks. My screen share here. Make sure I picked the right one.
We see coral.
Yeah, that's the
okay.
Presentation started. That's the screen it should show up on. There we go.
Okay, great. It's all yours.
Okay, so by now, many of you are at least somewhat aware of the research that happened this year, especially since it was largely due to our WABA community efforts. So for this presentation, I wanted to walk you through the discoveries that we made, especially after we started questioning what we thought we knew and how all of this culminated in some exciting results. And I have a lot to cover, so I'm going to talk at a pretty quick clip, but hopefully we'll get through it all in an hour. So Celastrina lucia was one of the few remaining species in Washington that I had not seen yet, and I was determined that this was going to be the year. So I emailed David James asking when he thought Lucia would be flying at Cowiche Canyon this year. And he said he and the girls hadn't seen any yet, but they'd keep an eye out for me. And then within a week, a small handful were seen and photographed by a few other people, but I thought I'd wait a little bit longer for better weather and to see if more would emerge. Then on April 19th, while exploring an unassuming little wayside a few miles south of Ellensburg next to the Yakima River, Dan and Kathy discovered several Lucia Azures and emailed John, Dave Nunnally, and David James. Then, since David James was aware that I was on the lookout for Lucia this season, he brought me into the group discussion that ensued. So, with only a few Lucia reported from Cowiche so far, and in spite of the forecast threatening rain that week, the temptation of Lucia was just too strong. So I took the rest of the week off work and drove over to Ellensburg that Wednesday. So 2 days later, but yeah, little did I know how that would shape the rest of my spring. So as soon as I arrived in Ellensburg, I picked up my dad. My mom still had to work that afternoon, and headed down the road to Bringer Loop. There were a few clouds, but as you can see in this picture, they were pretty scarce. And in spite of the wet forecast, it was actually quite sunny and warm enough for butterflies. And also, I wanted to point out, if I can find my mouse here, the little bits of green that you can see in the background in the underbrush, that's the red osier dogwood just starting to leaf out. That'll be important later. Okay, so to orient you to where this is, Ellensburg. So if we're looking at the map here on the left, Ellensburg's in the upper left corner, and they've got Canyon Road coming down and the freeway going through the valley here. So you head out Canyon Road, and just before you get to Thrall Road at the mouth of the Yakima Canyon, Ringer Loop is right here. And the Yakima River is flowing where those the dark green band of trees is. So this white box is zoomed in on the right here. The white line is Ringer Loop. Ringer South is a BLM access site on the Yakima River that's primarily used by boaters, but also includes a short loop trail through a tangle of red osier dogwood, wild roses, willows, and cottonwoods. It's a great place for bird watching and turns out to have a fair number of butterfly species, as I found in my return trips this year. It's pretty much exactly 5 miles south of Ellensburg on Canyon Road, and the middle part of Ringer Loop has actually been eroded away by the Yakima River. So that's been permanently closed where I marked the red X's here. So that can be a little confusing when you're looking for directions to get here. Some people have come down the north end of Ringer Loop and just get to the little parking lot and think that's it. But you actually need to come all the way down here almost to Thrall Road and get in here, and there's a great big parking lot, and then that little loop trail there. So I'd heard that Lucia in Cowiche and Bear Canyon are usually only seen in small numbers, often no more than a dozen or two on a given day. So I didn't expect to see very many at Ringer Loop. And I didn't see any for the first few minutes, just walking up the trail. And then I finally spotted one and caught it, thinking that that might be my only chance of seeing one. I was really excited. So the picture here is the very first one that I caught. And of course, my dad took a picture of me with my first Lucia Azure. But then a little bit further along the trail, we came into this big open clearing that this background picture is of, and there were azures everywhere. It was just glittering with azures all over the bushes. They were flying up and down these corridors, and then there was little side trails over to the river, and lots of osier dogwood and wild roses. And I noticed that they were landing on a big willow bush that's just out of the sight of this picture. And they seem to be nectaring on the willow catkins. I counted at least 30 or 40 in that spot, and then around 20 along the rest of the loop. They were all fresh males except for 2 females. And I collected both of those females and a sample of the males. Then emails began flying back and forth between me and John and Kathy, Dan, Melanie, Gary Bernard, and a few others. And Melanie wound up visiting the site multiple times over the next few weeks and documented more females as the flight season progressed. So why is this a big deal? Are you guys seeing this little, sorry. Is that screen sharing thing showing up on your screen? I don't think so. Sorry for the interruption. Currently, Lucia Azures are believed to range from Alaska, across Canada, around the Great Lakes and into Pennsylvania and New Jersey, south down into the Virginias. The occurrence of Lucia in Washington, so far removed from the central to northern British Columbia population, has long been a puzzle. We call it the Central Washington segregate to recognize it as a geographically separate entity, but we know so little about it that it's never been described as a separate subspecies. So Ringer Loop was just the tip of the iceberg. It caused me and others to question everything we knew or thought we knew about Lucia, building on the fragmented data that we had to that point. So the observations that we made this year exponentially increased what we know about this butterfly in Washington. But before I go into those details, I want to take a step back to look at the history of azure species in North America and how we've arrived at recognizing Lucia in Washington. So as of 1961, all North American azures were considered a single species called Celastrina argiolis, with several subspecies. Then in 1964, Newcomer, in his book, The Butterflies of Yakima County, stated that two forms occur, one typical of subspecies Echo, and the other with the dark markings as in subspecies Lucia, and both may be taken at the same place on the same day. So he recognized what we see today in Bear Canyon and some other sites in Yakima County, but just not at the same rank of each being a full species. In 1981, Celastrina argiolis was split into two species, Ladon and Nigra. And then in 1999, Dave Nunnally discovered the now famous Cowiche Canyon colony of azures. At the time, no one was sure what to make of it. Celastrina Ladon Lucia, as it was known then, was not recognized south of central BC, but the butterflies in Cowiche Canyon look very similar to those Lucia. In their 2001 book, Butterflies of British Columbia, Guppy and Shepard maintained Lucia as a subspecies of Celastrina ladon, as did other sources at the time, all of which considered Lucia's range to only be in Canada and parts of the Northeast US and Alaska. Guppy and Shepard adopted Celastrina Echo for all Pacific Northwest azures. That was the first time it was elevated to species. But there were difficulties regarding the form that Newcomer called Lucia. It's impossible to have two subspecies flying in the same location at the same time, so it didn't make sense that these darkly marked forms could be a separate subspecies of Echo while also flying together with Celastrina Echo, subspecies Echo. That's a lot of echoes. Unless they either represented the occurrence of Lucia as a separate species or possibly just an east side variation of subspecies Echo. Case in point, Butterflies of Cascadia came out shortly after in 2002, in which Bob considered Echo a subspecies of Argiolis and the sole Celastrina in Washington, but mentioned that there was a lot of uncertainty around the taxonomic arrangements and that restricting Lucia to Canada didn't seem quite right considering the Lucia form was so widespread in Eastern Washington. Then by 2001, Andy Warren and others were beginning to recognize Lucia as a full species, Celastrina Lucia, and that it occurred in places outside the previously recognized range. Andy visited the Burke Museum and determined that Washington had both azure species, Celastrina Echo and Celastrina Lucia. And besides the already recognized colonies in Bear Canyon, Cowiche Canyon, and other sites in Yakima County, Andy pulled out a few specimens from Kittitas County as well, three of which I show here. The one on the left is a male collected from Wilson Creek in 1966. And then the two on the right are females, one from Robinson Canyon in 1967, and the other from up by Cleellum in 1972. And you can see they all have that typical dark patch in the middle of the hind wing, the underside of the hind wing, but we'll talk more about that later. So in Andy's 2005 book, Butterflies of Oregon, he stated that Celastrina Lucia, or something closely related to it, occurs along the east slope of the Washington Cascades, but had not been recorded in Oregon. However, the azures in Northeast and North Central Oregon showed super variable ventral coloration and markings, and might be Lucia or another species such as nigrescens, which was currently a subspecies of Echo with an uncertain distribution. What to do with the rest of the dusky Echo forms or Lucia-like forms elsewhere in Eastern Washington was also still up in the air. So I'm not going to go into all the details of his observations and reasonings, but in summary, the taxonomy of Celastrina in the Northwest was still highly uncertain. And aside from clearly recognizing Echo in most of our area and Lucia in central Washington, these uncertainties have persisted to this day. Currently, the Pelham online catalog of the butterflies of the United States and Canada recognizes 9 species of azures, and only the first 2, Lucia and Echo, are believed to occur in the Pacific Northwest, with continued uncertainty about subspecies. So the central Washington segregate of Lucia, what I will also sometimes refer to as the extreme form of Lucia in this presentation, is considered to be restricted to Yakima County and a few historic records in Kittitas County. And outside of those recognized extreme Lucia colonies, all other Eastern Washington azures with dusky ventrals, either with or without the large discal patch, are considered to be Celastrina echo Lucia form. So this is where we were at the beginning of this year. And now I figured we could take a break for questions after all that taxonomy.
Okay, that's a lot of information there. If anybody has any questions, please go ahead and type them in. I think Caitlin's done a great job of laying out how messy this is with uncertainty about subspecies and species and different forms. If there are questions, if it starts to get to be just too confusing, we'll have another. I can call another break here later on. So please don't hesitate to type something in the chat if you're confused. There are some technical issues here, and if you have a question, other people may as well. But I don't see any questions, so why don't you go ahead and keep pressing on, Caitlin?
Okay. Actually, I'm seeing a thing that there's 5 messages in the chat.
Oh, now I'm seeing, sorry. Okay, you see them. What was the state of Celastrina in the eastern US?
Far more complex, and is still very complex. Yeah, it's a real mess. They've got, you saw that in the Pelham catalog, there's currently 9 species of Celastrina in North America, and we only have 2. So those other 7 are elsewhere. There's, off the top of my head, there's at least 1 in the Southwest, so like Arizona, New Mexico, down into Mexico. Our echo gets all the way down into California. And then I think there's a species that's kind of like Utah, Colorado, central area, Kansas, somewhere in that area. And then everything else is Eastern US, and is quite the jumbled mess that they're still trying to figure out. So.
Okay, there's been a little bit of discussion about dates. Dan just said that I think he and Kathy went to Ringer Loop on April 16th. I think your first visit there was April 21st. I guess one of the things I do want to comment on is just how extraordinary it is that a professional such as yourself was so connected to the amateur community. You heard about this amazing discovery so quickly. In any event, it was just days.
Yeah.
Another question, can you repeat what you said regarding the Central Washington aggregate? All other individuals are considered Celastrina echo Lucia form. Can you, I know it's a little confusing, if you could just go over that one more time.
Yeah, it's, so it's actually a segregate. A segregate is basically something that we recognize as fairly distinct, like distinct enough that we can describe it, but we don't know enough about it to be able to call it a full subspecies. And it's pretty much always a geographic entity. So like Central Washington segregate of Lucia. It's describing something that occurs in Washington that we're just not quite sure how it fits with Lucia. The dusky forms of Celastrina echo, sometimes people consider them nigrescens, but nigrescens is believed to only be the eastern edge of Washington, so only like around Spokane into Idaho and also in the Blue Mountains. So it gets a little sticky because these dusky forms occur outside of the normal nigrescens range. So we've basically just taken to calling them a Lucia form because we didn't know what to do with them.
And were you able to do any dissections of genitalia on the butterflies at that point?
No, and John is probably laughing because he's always trying to get me to learn how to do that, and I still haven't learned. So one of these days.
And he's John Pelham just chimes in and says genitalia are not useful here. So
okay.
Okay, if that's it on questions, we can make another break if people do have questions. So don't hesitate to type something in. So why don't you go ahead, Caitlin?
Okay. So back to that day at Ringer Loop, I started thinking about the habitat and piecing the puzzle together. Lucia were only known to use red osier dogwood in Yakima County, and apparently that's what they were after at Ringer Loop, but it grows in many places in the Kittitas and Yakima valleys and foothills, so why wouldn't Lucia be in similar habitats nearby? I was determined to figure this out, so I explored the area along Wilson Creek, east of Canyon Road, southeast from Ringer Loop, where this picture was taken. And I found 3 Lucia Azures here. So on this map, the big circle is Ringer Loop, and then these 3 circles down here are around the McCabe Fishing Pond and Wilson Creek. So then after I found those, I headed north up Canyon Road to Woodhouse Loop, and there's some ponds there and a little bit of state land. And so I went wandering back through the bushes behind the ponds, and sure enough, found another Lucia, and it stopped to feed on a willow catkin, same behavior as Ringer Loop. So the next question was, why are these colonies in Yakima and Kittitas counties so far apart from each other? It's trying to figure out what connects them. So this map is showing the yellow triangles are the known Celastrina Lucia Central Washington segregate records as of the time of this work. So in the red circle, these are my new, well, not all mine, Dan and Kathy made that discovery at Ringer Loop. So we've got the Ringer Loop one, and then the Wilson Creek area and Woodhouse Loop. And then these other historic records in Kittitas County from the Burke Museum. And then all the Yakima County records. You've got Cowiche Canyon and Bear Canyon and some other sites up in the hills there. So while several of these seem to indicate a pattern of forested foothills as kind of their typical habitat, the few in central Yakima and the addition of these new Kittitas County samples started showing a new pattern. And it suddenly hit me that they all are tied to drainages that are part of the Yakima River Basin. Cleellum is near the headwaters of the Yakima River. And then Robinson Canyon, Reeser Creek, Wilson Creeks, those all flow into the Yakima River. Bear Canyon drains into the Tieton River, which flows into Natchez. Cowiche Canyon, or yeah, Cowiche Creek flows into Natchez River, which flows into the Yakima. So while discussing this revelation with John, we hit upon another, wondering what if Lucia has been hiding under our very noses in other places, such as Azure Corner at the bottom of Reeser Creek. A lot of you are familiar with that location. It's up here in the northern part of the county, where up until now we've been calling it the Lucia form of Echo Azure. We weren't counting it as the actual Lucia Azure. So I started thinking that if all these Lucia colonies share similar habitat of osier dogwood and sagebrush steppe riparian corridors, perhaps that is the missing piece of the puzzle. After all, these dusky forms of Echo Azures only seem to occur around forest edges. The habitat transition from sage steppe lowlands to forested uplands. Azures further up Reeser Creek are the typical Echo Azure with no Lucia-like characteristics. So I started questioning that if this Lucia form is a normal variation of eastern populations of Echo, why do we not see that form in higher montane habitats, even in close proximity to places where we do see it, like at Reeser Canyon? Seems like you would have seen more of a grade. So I started wondering, could it be that these are actually a mix of Echo and Lucia, possibly with some hybridization creating intermediate forms? You see this picture on the left, Melanie photographed in 2020 and was questioning that as Lucia. And John and I really couldn't turn it into anything else. So then this discovery this year really helped tie that in. And then also I have this picture from the Sinlahegan Wildlife Area that, as you can see, it also is very dark and looks a lot like my ringer loop samples. So one more piece is that the genetics work had already proved that Echo and Lucia are they're separate species. So it's not a case of steppe and montane phenotypes, aka habitat-induced variation of the same species, like drier, warmer climates that might trigger certain markings and things. But we needed more data. We need to figure out what's going on here. So first, let's study the key characteristics of these butterflies. This is my sample of Lucia from ringer loop. And you can see there's a little bit of variation, but all of these males have really dark patches in the middle of their hind wings, except for the one on the bottom left is a little bit smaller. So that's the first thing that stands out. Some other things to look for is I started noticing that on the dorsal hind wing of a lot of the males, they have this white kind of a frosty band that almost looks like it's just normal wear on the butterfly. But when you look at them, you can see it on a lot of the individuals. There's just that kind of light white frosty band right there that you don't see on Echo. Also, they have really dark fringes. So they're either solid dark gray, almost black, or they're heavily checkered. So you see there's kind of smudginess in between, but a little bit more checkering on a lot of these. They have a really dusky gray ventral in addition to these dark charcoal markings. And then also the margin of the ventral hind wing is frequently solid gray. Sometimes it has more of the chevron pattern, like this one on the lower left. You can see the little bits of chevrons and then the dot in between those. But even when they have that pattern, it's still really dusky. I have taken to calling it smudgy. It's a smudged margin. And then the other thing that I noticed is that in this next band in, you again see a lot of white frostiness. And in some cases, it almost starts looking like white arrowheads kind of in between the wing veins. You just see these little smudges of white on there. It's kind of interesting. You can see that on the female here too. So these two on the right are the females, and you can also notice that this lower one has these big black patches in the middle of each wing, which was really interesting. We were questioning if that might be an aberration until Melanie went back. She went back multiple times, and I don't remember if it was about a week later, I think she photographed a female that looked just like this, had the exact same dark patches on the wings. So comparing with Echo, we've got the top row is our typical Celastrina Echo, and then the bottom row is just some of the pictures from the previous slide for comparison. So you can see that these are a lot more of a whitish gray on the ventral. And the other thing I forgot to mention on the previous one is the blue on these Lucia tends to be a little bit lighter. It's kind of an icy blue, a really bright blue. And the females are kind of a silvery, almost greenish blue, and extends out a lot more in the wings compared to this Echo female, where it has a lot less blue and it's really smudgy and gray around the wing edges. And then the markings are a lot more delicate, like all the chevrons on the wing margins are pretty thin, and they either have solid white wing fringes, like the one on the left here. They'll usually, Echo will usually have just a little bit of dark fringe towards the apex of the forewing, but the rest of the wing fringes, especially on the hind wing, are white. And then when they are checkered, it's only a really faint checkering. So where the wing vein comes in, there's just like a thin line of charcoal in the fringe. So those are the key characteristics to look for. So let's recap what we know at this point and how this set us up for research over the rest of the season. First, we know that Celastrina, Echo, and Lucia are genetically distinct species. Second, what we know as Lucia and Luciform azures in Washington are intermixed with pure Echo on the east side of the Cascades, but are usually separated geographically. There's only a few places where they seem to kind of mix. I'll get into their life history a little bit more later, but where they mix, these Lucia seem to exclusively use red osier dogwood, while Echo in eastern Washington almost exclusively use ceanothus or ocean spray, only occasionally using osier dogwood, although they do have a wide variety of larval food plants recorded throughout their range. So, and then the extreme expression of this form, what we call the Central Washington segregate of Lucia, is primarily seen in lowland sage steppe riparian habitats, while azures with markings ranging from that extreme form to those looking like Echo with just a little bit of Lucia markings occur widely from southern BC all the way into north central and northeastern Oregon, and also east into Idaho. So based on all that, we have 4 possible hypotheses. We know that Lucia is genetically distinct from Echo, but what we need to determine is if what we're seeing is both species. In other words, does Lucia occur in Washington, or is there another explanation? So if Lucia does occur in Washington, we have 2 options. The first is that it only occurs in a few isolated colonies, such as Cowiche Canyon and Ringer Loop, and all the other intermediate Lucia forms are Echo Azures with some historic introgression of Lucia genes. So they just throw some Lucia-like patterns. In other words, Lucia might have occurred more widely and hybridized with Echo in the past, but is now restricted to those isolated colonies, and the remaining Echo just still express some of those genes. The second hypothesis is that Lucia occurs across a wider range than previously thought, and that all these widely variable Lucia forms are in fact Lucia, while Echo is only made up of the lightly spotted form we refer to as pure Echo. There may be some occasional hybridization between the 2 where they overlap, but for the most part, they maintain distinct genetics and life histories. So then if Lucia does not occur in Washington, we have 2 more options. One is that all of these extreme Lucia and Luciform azures in Washington are actually just ecological phenotypes of Echo brought on by as yet unknown factors such as warmer temperatures or drier conditions in these sage steppe habitats in eastern Washington, or possibly by some historic introgression of Lucia so that some Lucia genes show up in eastern Echo populations, even though Lucia has long been gone and currently only occurs in the historically described range from Alaska through Canada and into the Northeast. US. Then finally, we must consider the hypothesis that if Lucia does not occur in the state, but these colonies exhibit all the characteristics of being independent from Echo, then perhaps this is a new undescribed species of azure. So until around 2001, number 3 was generally believed, as I discussed earlier, but there were a lot of unanswered questions that didn't quite fit. Then by 2005, people were somewhere between number 1 and number 4. Then the spring is when I started questioning all of these and putting more pieces of the puzzle together. And I started leaning more towards number 2, that Lucia has a wider range and that all of this stuff might be Lucia. So now with all of this in mind, let's see how the rest of the season unfolded and find out if we're finally able to solve the puzzle. So to answer these questions, my goals for this year were to determine the full extent of Luciform records across the state, which I could easily do by reviewing photos and plotting the records with my mapping software. My second goal was to try to collect larvae from Ringer Loop and compare them with the well-documented life history of Echo Azures. And lastly, I wanted to check for wild second brood. James and Nunnally had documented a small percentage of their Cowiche Canyon Lucia emerged as a second brood and exhibited a different appearance in their markings. No one had documented a second brood in the wild other than one of the Burke Museum specimens from Kittitas County had a later date that seemed to fit with the second brood, and its appearance was kind of similar to what James and Nunnally found. So considering that the Ringer Loop colony seemed so much bigger than the known colonies in Yakima, I figured that that was as good a chance as any of finding these elusive second brood little butterflies. With everyone now on high alert, Cheryl Boleen soon reported some possible Lucia Azures from near Moses Meadows. This was in Okanagan County on May 16th, and she found these in association with red osier dogwood, as you can see in her picture there. Also, you can note in the photos, you can see the very heavily checkered fringe on all of those butterflies. And the two on the bottom have that solid margin, the ventral margin that I was showing. Even though this one on the bottom right, it doesn't have much of a solid discal patch, but it definitely has a really solid margin. And then this one in the upper left, it has the big discal patch, but it doesn't have a solid margin. There's still those chevrons and dots, but they're a lot more thickened and dusky compared to typical Echo. Also of note is that Cheryl did not see any pure Echo forms at this location. Meanwhile, I took a second look at all my Celastrina specimens collected over the years, found out that I really didn't have very many because they're so common, I just never bothered to collect them. And I realized that I had some from Azure Corner in Lower Research Canyon from 2005, 2 from Horseshoe Lake in Pend Oreille County from 2018, and some from the Sinlahican Wildlife Area in Okanagan County from 2013 that all resemble my Lucia sample from Ringer Loop. Several of these have the large discal patch, and some have that solid ventral margin, while others have a heavily thickened margin, and all of them have dark or checkered wing fringes. And with these 2 on the lower left, I wanted to point out that even though the discal spots are fairly small, they don't have that big patch like a lot of these other ones. The margin is still really heavily smudged, very dusky, and especially on this one from Research Canyon, look at all that white in that band there. You can kind of see those white arrowheads and just a hint of them on the Sinlahican specimen. Other Lucia candidates from the northeast part of the state are seen in these photos by Jean Damerel and John Bauman, taken between 2011 and 2013. Note the solid margin on 2 of these, one on the lower left and in the center here. And then the checkered fringe on all of them. It's a little harder to see in John's picture because the sun is shining on it so brightly, but you can see it a little bit better on the upper part of the forewing and lower part of the hind wing. There's a little bit of gray in there. And then it's got that big patch on it. And then Norbert Kandla has noticed the same trend in Southeast British Columbia for decades, compiling several examples of specimens and wild photos over the years, such as the sample shown here. Obviously, the top row are these weird little dusky form, Lucia things. And then the bottom row are what we would call pure echo. And again, note that even when this individual doesn't have a big discal patch, it still has a very smudged margin, very heavily checkered wing fringe. And even the discal color, the ground color is a little bit more of a brownish gray than these echo. Okay, so now let's take a look at the larvae. On May 28th, I revisited Ringer Loop with my parents to search for larvae on the dogwood flowers and seed pod clusters. We found around 20 third to fourth instar larvae, 12 of which I brought home to rear. Some of the third instar larvae had faint cream markings, as you see in the first 2 pictures. But once they reached the fourth instar, the 2 on the right, they were all solid green with a pale lateral stripe. Lateral is on the side here, this pale stripe here. One of them had a dark rose brown patch above the head, see in this picture. And then one had more extensive rose brown markings down the back and sides. All of these turned a medium brownish green when they left the flower clusters and started searching for a place to pupate. And then they lightened to this really amazing translucent blue and beige color before pupating. And then the pupae were kind of a pinkish brown with lots of black speckles, which darkened even more over time. So some of them are almost black now. Then a day after collecting those larvae at Ringer Loop, we drove around to the Nannan Basin just to explore and look for whatever butterflies were around. We weren't really planning to look for azures. And Nannan Basin is a little tricky to get to because Nannan Road comes up out of the valley, but there's a gate at the end of it. So the only way to access the basin is you have to go up either Schnebly Road or Coleman, I think. Yeah, I think it's Coleman Canyon, and get up there through the network of roads and drop down into the Nannan Basin from the northeast. And then you can drive, there's a road that goes right along the creek all the way down until you hit that gate again. So there was still a whole bunch of snow higher up, and we weren't seeing any butterflies until we dropped down into the basin. We started seeing more and more. And when we got almost to the gate, we came into this area along the creek where there were huge red osier dogwoods all along there, and started seeing azures. Mom and I found 2 unhatched eggs and 1 hatched egg that you see in this photo here on one of the dogwood flowers. And my dad was busy scanning the bushes on the lookout for more. It was getting towards the end of their flight season, so they were all pretty faded and tattered, and a lot of females. But I got a really interesting range and variation here. So you can see the bottom 3 are pretty typical echo. And then these ones on the top look very similar to the Ringer. I'm going to do that a lot. The Ringer Loop butterflies. And then these 2 here are kind of intermediate. So this one, again, it's really faded. So it was kind of hard for me to decide which one I would put it with. I think I'd put it up with these Lucia forms because it does seem to have that solid margin and a little bit of white frostiness on the wing, but the spots are really small, like a typical echo. And then this one has a little bit of that patchiness in the disc, but it has a little more of an echo-like margin. Oh, and then also what I found was interesting is the setup seemed to be just like Azure Corner at Research Canyon, because it's right where the lower valley is coming up to meet the montane area, getting up into the forested hills. And there's this big steep slope with a whole bunch of ceanothus on it that comes right down to the road. And then right on the other side of the road is all these osier dogwood bushes along the creek. So it's perfect place for these to meet. So then on June 17th, I examined the location at the San Mohican where my specimens and photos were taken. And it struck me that it's very similar, again, to the Azure Corner and Manum Basin situation. It's partially up a canyon where the open steppe transitions into forested habitat, and a small tributary drains down into the San Mohican Creek. And right in the middle was a big red osier dogwood bush that I had never noticed there before. There's a lot of red osier dogwood down along the San Mohican Creek through the valley floor, but very little once you get up into the trees higher up. The dogwood down in the valley already had large seed pods. It was well past the stage when I found larvae at Ringer Loop, but this dogwood had just finished flowering, and I immediately started finding larvae. So you can see this teeny tiny little one on this dogwood seed here. So on the next day, I returned to that spot with Dale Swedberg and Cheryl Boleyn this time, and we found several more second to fourth instar larvae with all of us searching, many of which were tended by ants, which was really cool. So I got some photos of that. You can see that they look identical to the Ringer Loop larvae. They're that solid green, velvety green with just a really faint lateral stripe. And this one had a little bit of rosy brown on the head, but most of them were solid green. The picture over here, you can see ant in the middle of the picture, and then a larva over here on the left of it, kind of behind the seeds. There's another ant in the back here, and there's another larva right here eating on that seed pod. And there's an ant. You can kind of see the abdomen of the ant there and its leg wrapping around the larva. So yeah, those ants were all over the place, very busily attending the caterpillars. So that was fun. One of the first instar larvae, so I wound up bringing, I think it was 12. Yeah, just like Ring Loop. I collected 12, but they actually turned into 14 because there were two little stowaways that were so tiny that I didn't even see them. They were on the seed pods that I collected to feed all the other larvae. And one of those little ones, as it got older, once it reached the third instar, it developed a stronger, creamy chevron pattern on its back that I didn't see on the other ones so much. And then this picture is what they look like when they leave the seed pods before they pupate. And then right before they pupate, they turn more that translucent blue picture, the blue color that I showed you previously. So this larva, I'm keeping it separate, the pupa that it turned into, I'm keeping that separate and overwintering these, and hopefully they'll emerge. Sometimes it can be a little difficult getting them through the winter, but I wanted to keep it separate just to see if the adult looks any different than the other ones. Now these are echo larvae. They're very variable. But one consistency is that they always seem to have a noticeable pattern on their back. So they have a lot of different colors that mostly seems related to what they're feeding on. Like I said earlier, they have a wide range of food plants. But even when they're mostly green, they have these two big dark green patches, kind of squares near their head. You can see it on this one, two big dark green patches. And then even when they're colored, it's got these two dark patches near the head, and this one up in the upper right corner. Again, those 2 dark patches. And then I also noticed that there's 3 dark spots near the rear. So on the green ones, it's really noticeable, those 3 brown spots. On the colored ones, you don't see it quite as much because they're spotted all the way along, but that last series does seem to be a little bit darker than the other ones. This spot on the upper left or upper right photo, that spot is a little bit bigger and darker. So if we go back to my previous picture and look at this one, you can see towards the head, it does have slightly darker patches, 2 patches there, but there's no hint of the 3 spots near the rear. Okay, so then on June 12th and 14th, 3 adults emerged from the ringer loop larvae that I had gathered, and they all had ventral patterns matching the 2nd generation phenotype that was described by James and Nunnally for their book in 2011. And the 3 out of 12, so remember I kept 12 larvae and 3 emerged as this 2nd generation. So that's 25%. James and Nunnally reported approximately 30% emergence, so roughly the same. And I stopped at Ringer Loop on my way up to the Sinlahican, which is the same day that these were starting to emerge, but I didn't see any wild 2nd generation Lucia flying around. But then on my way back home on June 21st, I stopped there again and wound up seeing 4 males flying around. I collected 2 of them, and then Melanie returned in subsequent days and wound up reporting a few others, including a female. But the numbers seem to correlate to that 25 to 30% of the total population based on our earlier spring counts, what we were estimating, the numbers that we were seeing. So the photo in the upper right is just a comparison of that first brood, what they look like, and then what this second brood looks like. So you can see the dorsals have an even stronger expression of that white band on the hind wing. Unfortunately, I took a chunk out of this one with my forceps that I felt absolutely sick about, but you can still see enough of it. And then on the ventral, they have a reduced discal patch, and they don't really have a solid margin, although this male does a little bit, but it's still really dusky. And then they have a lot of white over the gray. And then this female is just amazing, absolutely amazing. Lots and lots of white on the dorsal hind wing, lots of white on the ventral hind wing, and really pretty blue. Then on July 8th, an adult emerged from the Sinlahican sample. It has some similarities to the 2nd generation ringer loop and Cowiche Canyon Lucia, but has a slightly darker dorsal and ventral ground color. It's a little more similar to the first brood. And it has an almost solid black fringe on the wings. And yeah, so that I only had this one emerge, all the rest of the pupae remained. So I've got them all tucked away outside. Hopefully they'll make it through the winter, and I'll report back and see what I wind up with next spring. Okay, so now what? After reviewing all this data and differentiating the Lucia and Luciform records from pure Echo, the map now looks like this. So these yellow diamonds are everything that we would consider Celastrina Lucia Central Washington segregate based on all the data we gathered this year, and looking at all the pictures, historical records, museum records, photos and all that. And then remember when I was talking about nigrescence earlier, these purple dots are what would be considered Celastrina echo nigrescence subspecies. And then blue is the echo subspecies of Celastrina echo through the state. So you can see all that where they overlap, but all the yellow diamonds are pretty consistent in this area. Let's see. So based on all of that, all of the data that we gathered this year, it showed so many similarities across Eastern Washington that it would be inconsistent to say that Lucia exists in only a few isolated colonies in Yakima and Kittitas County. So therefore, we can eliminate our first hypothesis. We can also say that the form is distinct from Echo Azures. It can easily be identified by adult appearance, and their larvae seem to be consistently distinct in appearance, and they seem to prefer drainage habitats adjacent to typical montane echo habitat on the east side, which therefore eliminates our third hypothesis that they're just all echo. So that leaves us with the hypothesis that I was leaning towards, number 2, that Lucia extends from Canada down into Oregon, or hypothesis 4, that all these dusky forms could possibly be another species that's not echo or Lucia. So after gathering all this data, John and I sent a few samples to Nick Grisham in Texas to run a DNA analysis to see if maybe these could be distinct subspecies of Lucia compared to the rest of its range. And here is what the Celastrina echo DNA analysis looked like. So this image overlay that you can't read is the whole structure file, and it's all capitals because this is the name of a particular computer software that they use. The columns on here represent the nucleotide sequences by color and the Z chromosome, which is a lepidopteran sex chromosome like X and Y in humans. And all of this is produced by this structure software, which is widely used in studies of population structure. Now, I can't explain exactly how it works other than the colors indicate groups of species or subspecies. So they're representing similarities in the DNA sequences. So the yellow that you see here is Celastrina echo, and on the left you can see all of these samples. The numbers are these the unique codes for each of the samples, and then the country and state and county where it's from. So where there's little bits of other colors, those are where some those represent some minor variations in the nucleotide sequences, possibly from introgression from other species or geographic variation, both of which may contribute to grouping populations with shared attributes into a subspecies. So like you can see little bits of blue here, there's blue elsewhere, lots of red and orange, but all of these where they're grouping, you can see kind of where the distinct species are. And then sometimes there's some other slight variations that might be another species, or it could just be a subspecies of that larger group. So here we have the 2 samples that I sent Nick. These were my pure echo samples. The first one from Okanagan County on the list, and the second from Kittitas County. So they were from Reeser Canyon, a little bit higher up than Azure Corner. And then from the Sinlahican at the same location where I photographed that other Lucia form and got all those larvae off of the osier dogwood. Oh, and then the other thing I wanted to point out was, even though these are a pretty typical echo, this one on the left from Racer Canyon, it has a little bit more of checkering on the fringe. So just want to point that they sometimes can look like that, but the rest of the markings are pretty thin, pretty delicate looking. And here with all the red and purple, we have Lucia. So up at the top, you can see Alaska and Yukon with a lot of purple. So those are definitely distinct, probably their own subspecies, if not their own species. And then you get into Alberta, Saskatchewan, Manitoba, Quebec, Wisconsin, New Jersey, New York, Pennsylvania, Maryland, Maine, Ontario, Nova Scotia, New York, Michigan. But where's our Lucia? I don't see him here. So we keep scrolling. And here we have a new species. All of our samples, including the so-called intermediate forms, are totally distinct from Echo and Lucia. You can see samples from British Columbia. These are Norbert Kondla's southern British Columbia samples. Yakima County is the Cowiche Canyon colony. We've got Columbia County in Washington down the Blue Mountains. Kittitas County and the rest of Kittitas County down here, those are all my Azure Corner, Reeser Creek, Nanum Basin, and Ringer Loop samples. And then Okanagan County, we've got 2 samples from the Sinlahican, and Pend Oreille are my 2 specimens from Horseshoe Lake. And I sent Nick both ones that looked like what we were calling Lucia and several samples of intermediate forms just to see if they were, I was expecting them to come back as hybrids or, maybe Echo, trying to figure out where the difference is. But yeah, this was just kind of blew our minds. Very, very, very exciting. So your assignment for next year is to start looking for Yakima in all the right places. I do ask that you keep this news fairly quiet for the next month or so. Just don't go posting about it on the internet until we make it official. John and I hope to publish within the next couple months. We're already well on our way writing the paper. We plan to call it Yakima in honor of the native spelling of the region that was key to solving the puzzle. So next April and May, keep an eye out for any and all osier dogwood that you can find in eastern Washington, even northern Oregon, Idaho, and western Montana would be helpful to get more data from as we work on sorting out the full range of this species. We're pretty sure it extends all through Idaho, so it'd be good to get some samples from there. Photos are great and really help in this effort, but we could also use more samples for DNA testing, especially of any that we've been calling intermediate forms. So if you find ones that you really can't quite place with Echo or Yakima, those would be good to sample to see if we can work out exactly where the cutoff is and how to identify these. And maybe there are some hybrids, where they're both in the same location and Echo is using red osier dogwood a lot. Inevitably, it wouldn't surprise me if we have a few hybrids here and there. So for DNA, we don't need fresh, pretty butterflies for DNA. Even a tattered one is usable as long as it was collected live and frozen and then dried all in a clean container to avoid any contamination from other butterflies or organic material. So in other words, a dead butterfly on the road or stuck in your grill isn't usable, and keeping multiple butterflies in a single container wouldn't work either. And then if you are going to do that, you must include latitude, longitude, date, and a general location description. And then send that to me or John and we'll get it off to Nick. So, and then one more. So that's the end of my presentation, but I had a picture I was going to show you guys. I can pull it up here. Not that one. So that is a picture of the osier dogwood growing along Reeser Creek, or sorry, Sinlahican Creek at the bottom of the Sinlahican Valley. And just today, I got this photo from a guy who spends half his time in Alaska and half his time in Washington. And he went to the Sinlahican in May, and he's been up in Alaska all summer, and now he's back and he's just getting around to sending me the photos that he took in May. And this was one of them. So this is the first documented Yakima in the Sinlahican Valley, because all the other records that I have are just from that one spot higher up. So that was exciting and very coincidental that I just got that today. So with that, I'm sure everybody's got a lot to discuss.
Well, thank you very much, Caitlin. This is just an astonishing piece of work, and kudos to you for seeing it through and seeing it through so quickly. I want to reiterate how astonishing it is to me, as someone who's only recently joined this community, that there is such a close relationship between professionals such as yourself and people who are just out there loving to be outside, like Dan and Kathy, and who alerted the professional community so quickly and brought this to such an astonishing conclusion. So I want to thank you, and please feel free to unmute, and we can have a discussion. You can give Caitlin your congratulations directly. Okay, so Jonathan says he would like to speak for a moment. Can you unmute yourself?
I have unmuted myself. Can you hear me?
Yes, thank you.
I would just like to say this about all of that. I was wrong. Everyone else was right. And I am so delighted, because science is always a matter of correcting and moving on. And the resolution that we have received this year, this single year, basically revolutionized how we look at Azures in our state, and in fact, in the whole country. So there you go. I'm done.
So that is just so terrific. And Danny and Kathy, you guys were the ones who kind of kicked this all off. So kudos to you for noticing. I know you're out there looking at birds, but for noticing these butterflies, knowing enough to have seen them in Yakima Canyon, presumably, and notifying the professional community. So you guys are just the best.
Well, actually, we were being pests.
You were being pests?
Yeah.
Okay.
I just always let me show Jonathan this one. Let me show this one. We got to show that one. And Dan saying, Kathy, you go show him, because I show how many times do we throw these at you, because wow, this is really cool. But okay, thank you for running with it.
Well, hey.
Yeah, if it hadn't have been for that, like I would have just been going to Cowiche Canyon to see Lucia, and we wouldn't be any further either. It's interesting how the whole chain of events happened too.
And I agree with the concept of the teamwork involved. I've been saying this to everybody I say across the country, to all my cohorts and everywhere, that we have this incredible community out here. They operate at many different levels, and they operate with complete, the complete satisfaction of the community. It's just a kind of an amazing thing. John Bauman knows it. He's been doing this for a while. Jean knows it. She's been cooperating for a long time. But every little level where people cooperate like this is a defining moment for us, and I'm just so pleased to be a part of it.
Yep, agreed.
Yeah, it also speaks to all the photos that people take and the specimens that we collect over the years. It might not seem like much at the time, but it all builds up, and eventually you get that one little piece, and you can go back and look at all of that stuff. Go look at all of John Bauman and Jean Damerel's photos from Spokane that they've been sending over the years, going, is this Lucia? Is this Lucia? So yeah, it's like everybody was kind of on sort of the right track. It's just like I said, it was a mess that nobody really knew what to make of it, and now we know. And we only got this DNA results. We just learned about this, I think 3 weeks ago now, like 2.5, 3 weeks ago. So this is very recent.
Caitlin, this is Dave Nunnally. Great work, really fabulous work. You have the perfect map up for my question that's coming up here. And I've already asked this of John, but there are areas where both Celastrina, Echo nigrescens, and the new Yakima occur together in the Northeast, also in the Blue Mountains. And is there any way to separate them other than doing DNA analyses when you do find them together?
I think we will be able to, because actually in those areas, it seems like, because there was a Columbia County sample that got sent in that came back as Echo, like pure echo. That was on this one. There's a Mason County. Yeah, so there's Columbia County. Oops, didn't mean to click. Columbia County, I guess actually on this one is just above the red line. So that was from the Blue Mountains and it came back 100% echo. And from what I've seen in all the pictures, that even if those are called nigrescens, they still seem to be a pretty typical echo. Like they might look more like this Reeser one where, you know, they might have just a little bit of a heavier marking on them, but pretty much everything that is what we've been calling dusky echo or whatever seems to be coming back as Yakima. It seems to make the most sense. So at this point, we're actually not really sure what to do with nigrescens.
Do you have altitudes on those observations up in the Northeast?
Some of them, like probably not for some of the photos, but John, you could speak more to that.
Oh yeah, dump it on me.
Well, you're the record keeper.
Well, first of all, I'm glad that she answered the question about nigrescens. I would say this: everything that we sent that had dusky markings, it would be Lucia formed by my, definition beforehand. They all came back Yakima. Everything that looked like an echo came back echo. So we may have to retire nigrescens altogether.
Yeah, I'm just wondering whether because the echo and the Yakima form do occur in the same places, just at different in different habitats.
Not necessarily. They actually they're concurrent in Bear Canyon, at Azure Corner. I think I could document at least 3 or 4 places where they co-occur the same day. Most of the time, Lucia or Yakima, excuse me, Yakima is earlier. That seems to be a constant form. But like if you go to Cowiche or Ringer Loop, you don't get any echo at all. Where they co-occurs with echo, there's a number of places where they're absolutely side by side. And we just may get to the point where you see something like this is up on the screen right now. You know what it is because that's what it looks like.
Yeah, well, in this Nannan Basin sample, that's kind of what I was alluding to here is these 2 samples in the lower, the middle here are what we probably would have dismissed as nigrescens, and so far, I can't remember. There were 2 of these that I sent in for DNA, and I can't remember which ones they were now. It might have been one of these 2 intermediate ones. I'd have to go look. But every one of them came back as Yakima, 100% Yakima. So we definitely need, that's only one intermediate sample from Nannan and 1 strong sample from Nannan. I had 1 intermediate and 1 strong from Reeser, 1 intermediate, 1 strong from Sinlahican. But everything was Yakima. So that's why we need more samples of these, just to see if we can carry that out and say, yes, anything that looks like this, if it just even remotely has that discal patch or that solid margin or that overall duskiness, that's Yakima. We can't 100% say that at this point, but I'm pretty strongly leaning that direction.
So Caitlin, this is Regina. I have a question about your host plants. You were talking about how the Lucia/Yakima is only on the red osier dogwood, and when they co-occur, the echo never is. Is that
no? Echo, like here at Nannan Basin, I actually observed some of these echo were ovipositing on the osier dogwood. It's just that echo will use osier dogwood. It's just that in these habitats where they co-occur, there's almost always ceanothus and ocean spray and other stuff there, and I almost always see them ovipositing on that. And then the larvae that I collected, Ringer Loop obviously, but Sinlahican, like all the larvae I got off of that osier dogwood bush at the Sinlahican all look like the Yakima larvae. They don't look like the echo larvae that I've collected at the Sinlahican. And I know that the pure echo also occur at that same spot. So it seems like if the echo are there, they're pretty much just using the ceanothus. You just might occasionally go, oh, well, there's this dogwood here. We can use that too. But
So might I ask if Caitlin, you and John and whoever else is involved at the research end of this could send us questions over here to the Far East that we can pack over into Horseshoe Lake, Hog Lake, Mount Spokane, Idaho Panhandle, and try to answer that stuff for you. What do you
Yeah.
need?
Any documentation that you've got out there. So, DNA would be helpful. And I don't know how much Nick will tolerate us for how long, but
I have a few
we're throwing things at him,
specimens of these species.
yeah. So I would say, trying to cast a wide net would be best. So, I already have those 2 Horseshoe Lake specimens that I collect. That was actually the trip I took over there, and Jean Damerel showed me around.
Right.
She took me up there, so it's the spot where she photographs all that stuff. So that's I would say that could be marked as covered. We could always do a couple more samples from there if we really wanted to, but other places, like right there by Spokane, I think you've gotten some photos from, there's like a wildlife refuge there. I didn't show all of your photos, but I was looking back through everywhere you've taken photos from. But just kind of get, data points from different places. It'd be good to get some stuff a little bit further up. Norbert's stuff is all from the Pend Oreille Valley and the Canada side, but it'd be nice to get some from just south of the border there too.
Carl Barentine asked a question earlier about a bug guide image from Cheney. And we're getting them, we have images we recorded from Hog Lake. Yeah, this is a steppe butterfly. Now, this is a butterfly. You don't find azures in the steppe most of the time. Well, with this exception. Of course, that's why we haven't found very many of them, because we don't go out in the steppe in April and May. But yeah, he had a bug guide specimen he mentioned from Cheney. I would call that a Yakima. Yeah. So you're going to find them, in places. Huh?
Oh, I was just showing the yellow diamonds on here.
Oh.
I can't remember exactly where Cheney is. Is that the yellow diamond down here?
Let's see.
Maybe?
Kind of southeast Spokane County. Southwest, I should say.
Yeah,
That's it.
probably somewhere between that yellow diamond and the purple dots.
So Caitlin, can you repeat how to prepare any captured Celastrina for your DNA analysis?
Great. Yeah, so if you catch any that you do want to submit for DNA, you want to keep it as clean as possible. So when I'm collecting, I use glycine envelopes, which on the table behind me. I won't grab them now, but so they're just, it's kind of like wax paper. So you don't lose a bunch of scales and stuff like that. But you could also just put them in if it's a clean jar, like a little Tupperware jar, some sort of specimen container, and then put them in your cooler until you can get back and put them in your freezer, and then let them dry out and send them to us and send them in the mail. So it's just all about making sure you keep a clean container, that if you've had other butterflies in it before, just make sure you wash it out really thoroughly. Because they get DNA out of the tiny little legs off of these butterflies. That's all we need, is just a leg off of these. So if they can get DNA out of something as tiny as these little Celastrina legs, then even just a tiny little chip off of another butterfly's wings could be enough to throw off the DNA.
Okay.
And then also, sorry. Well, when you do that, just make sure that you maintain really careful data on that. So either like when I submit the records, I just write like a little code number on the vial that has the legs in it that I'm sending Nick. And then I associate that code number. I make sure it's a unique code on each of them. And I associate that with a spreadsheet that has all the full data in it.
Okay, Gary is also asking, is material in RNA later solution acceptable? I'm not sure what he means by that.
I know what he's talking about, but I'm not sure. John, do you know?
No, that'd be fine. Yeah. I mean, Nick is an advanced genomicist, and so he can take specimens that are 100 years old and extract DNA Yeah, he can make do with that. RNA, you have to, Caitlin made the point that you have to protect against, extraneous DNA. So once you dump it in that solution, you're pretty much secure. So that's fine.
Okay, we also have a question from Nixie Kibele about your emails. Are you willing to share your emails with Nixie?
I gave her mine already.
Yeah, I didn't put mine on a slide or anything. I guess I could do that.
Can you just tell me and I'll type it in right now?
Yeah, so it's my name, CAITLIN LABAR @gmail.com.
At gmail.com. Okay. All right. Let's see, another question from Melanie. She's asking, am I understanding you to say that Washington has no C. Lucia, only Neko, Negrescens, and Yakima? And you're free to jump in too, Melanie.
Yep, we do not have Lucia in Washington. So the Canadians have been right all along that Lucia only occurs from Alaska down across Canada. It only gets into central BC. It doesn't even get into southern BC.
Mind-boggling.
Yeah. So yeah, I've kind of been hitting it's yep, it is all one species. It's just not Lucia.
Wow.
And it's amazing how distinct it is too. I mean, you can see that blue, just it's totally different from all these other. So when somebody had asked earlier about the other Azure species in the rest of the US. So that's where a lot of this other stuff comes from. And like I said, it's really muddy. So you look at this and you see all this red. Lucia is all this red and a little bit of purple. Actually, I can figure out, sorry, I've got like 3 screens here and I can't find my mouse half the time. I'll pull up that diagram on full screen so I can zoom in.
So Gary is asking, just to be absolutely sure, there's no Yakima west of the Cascade Crest then? No Celastrina Yakima that we know of?
No, not west. What west of the crest is all the pure echoforms. We have not documented any Yakima. There's a couple records from Klickitat County that are a little bit suspicious, that I want to investigate that area more next spring. But
And that would be because there's no SH step. Is that basically?
well, no, there is. It's just one of the records we weren't sure about the location because it seemed like the location given might not have been actually where it was taken. So we're not 100% sure it was from that part of Klickitat County that it said it was from. But yeah, so I think there's only like 2 or 3 records from Klickitat. So I want to investigate that a little bit more. And then, we've got those records from up by Cleellum, and then I think Ben Mouse got some from maybe even a little bit closer to Snoqualmie.
No, that was Erin Robinson. She sent me 2 images on her cell phone from Gold Creek near the pass. Okay, and they were clearly Yakima. And that's one thing I need to make clear. Yakima can penetrate into forested habitats. Okay, it's not. Yeah, Echo can't penetrate into steppe habitats. That's the clear thing, is that you're out in this open. I mean, some of these habitats are open steppe. You look at Cowiche Canyon. That's a desert canyon, and that's the hallmark of Yakima, its ability to pioneer these shrub steppe habitats.
Yeah, and I think that's one of the reasons why we haven't figured this out yet, is because if they're out there in these shrub steppe environments, and they're one of the first things to emerge, the only time us butterfly folks are going out there is to look for green hair streaks in March, and we're generally not tramping along riparian corridors that don't even have any flowers or leaves or anything, and there's no green hair streaks down there. So we're not in the right place to see these. They're pretty restricted to those those habitat areas. So there was I don't remember the date, but there was a record a photo that we found that was over in the coulee area. I think it was north of Dry Falls, it's further up from that. But yeah, early April, we got to get out there really early April. Like what I was seeing at the San Mohican when I was there in mid-June, mid to late June, and the larvae were still, they were on that osier dogwood higher up in that side canyon, but all the dogwood, and it just finished blooming, but all the stuff down in the valley was like full seeds. They were all already starting to turn white. It was well past when the larvae would be on there. So, and like that guy that sent me the photo today, that was, I think mid-May when he photographed it, and that was a female. So that would be mid to towards the end of their flight period. So we're talking early May at least, for out in the open habitat areas. And then Ringer Loop was, April 16th to April 21st and further, it was probably like a 3-week flight period. So you can get out in the coulee area, out in the basin, try to find some riparian habitat that you can access in early April. That's probably the time to be out there.
Miss LaBar,
What?
read the title of your paper again, your presentation.
Looking for Lucia in all the right places?
We now know to look for Yakima in the right places.
Mhm.
So can you all talk a little bit about how you chose the name? We're all familiar with the spelling of the city, but you've chosen an alternative spelling, and some people are familiar with that. But I thought for those who aren't, you might talk about that a little bit.
Yeah, so originally, John and I were planning on describing a subspecies of Lucia because we were really excited with all this data we were gathering, and it was finally, just exponentially increasing what we knew about this Central Washington segregate. So we decided we wouldn't need to call it that anymore. We had enough evidence now to be able to define it as a subspecies until we got the DNA back and found it was a full species. So we had already decided on the name Yakima for the subspecies, and that was, I came up with the name just because of, it was shortly after my breakthrough of, wait a minute, these are all like drainages all connecting into the Yakima River Basin. And we hadn't really thought about it as in a bigger area at that point, but that was just the first name that I threw out. I thought, well, we could call it Yakima. I mean, it's an easy name. Everybody is familiar with it. It'd be an easy thing to call it. And John suggested calling it by the native spelling. So John, I don't know if you had any other thoughts as to that. I don't know your whole thought process, but that's kind of how it came about.
I went to school at Yakima Valley College, and the tribe would have me out and visit them. So like it's a personal homage to them, That's the way they spell it,
Hi, Caitlin. Congratulations.
Hello.
Jim
Thank
Ray from over here in Clickitat County. I will keep my eye out here and go through my specimens and find out how many of them that I got and didn't know what I got.
Yeah.
But anyway, I hang out with the Yakima quite a bit. And so basically their rule of thumb is, if you're talking about the place, Yakima should have an I in it. If you're talking about the people, it should be the all A's.
Okay.
I don't know. That's the rule of thumb that they went by.
Mhm.
And I think it works fairly well.
Yeah.
What if you're talking about a butterfly?
You can call it whatever you want.
Well, the other thing too, I thought, after we started learning more about this is that, and then I was like, well, if it's also in BC and all these other places, that doesn't really say that it's a Yakima Basin butterfly. So maybe we shouldn't use that name, but then, but we're kind of stuck, we were attached to it at that point. And also I was thinking that, the original land of the Yakima tribe was, it wasn't just the Yakima Basin, it was a little bit bigger than that. So I thought, well, it's still close enough.
Well, it's common to name species after a person, so maybe this is a
Yeah.
special situation, naming it after a tribe. I think that's pretty great.
Well, there's a number of taxa that have been named after indigenous people. Shoshone, for example. That's not a big thing. I like it. Yakima. It's a nice euphonious name. It's Northwestern, and the butterfly largely is a Northwestern butterfly. We consider Southern Canada, the part adjacent to us, Northwest. I mean, if we consider Western Montana Northwest, we certainly can do that, right?
Mhm.
This is getting a little bit far afield, but I did want to say I know that we had a discussion not too long ago about working more closely with the tribes and engaging them more fully. And I began to think that there's a college in Toppenish that's called Heritage University, and they have a biology program. I wondered if it might be worth connecting with them and offering to go over and talk with them.
Yeah, when we get that paper published, I might, take it on down there and, talk with them. I have connections with the Rainwaters and Dollar Hides, so I might be able to make something happen. They're very jealous of their land. They're proud of it, and so like if you give them a reason, name a butterfly after them, they could probably get into that. I certainly nothing wrong with trying that. And it's probably all up and down. One of the things is we're starting to realize it's probably a lot of places that people have looked at for years and years. It's like Caitlin said, I have been to Ringer Loop myself in July. So I was sitting on top of this whole thing. Well, Caitlin, why don't you tell them how far away your folks live?
For crying out loud, it's less than 5 miles away from where I spent like 14 years of my life. It's literally right down the road. I mean, yeah. So it was a little embarrassing. Like, how did I not know this place existed?
Well, this is all really terrific. Anybody else have anything they want to say or comment on or question? I think everybody's stunned, to tell you the truth, Caitlin. We're all just like, oh my gosh. A new
Gary
species.
Bernard,
Everybody's
this is
gonna
just
get off and go
wonderful.
start listening.
Cheryl, did you want to say something?
No, this is great. Good job.
Well, thanks for being a part of the whole story.
Yeah. You were finding half of the larvae we were looking for. You're a big part of it too.
So are you going to send off that Moses Meadows sample?
Yeah, it's been sent off already. He just has, look, he's a busy man, and doing sequences is hard, and we can't do it. So it's on his schedule, Yeah, we'll get it back, and it's going to be a Yakima. Don't sweat it. I call all the stuff in the Chihuahua Yakima, and it surely is. It saves us, look, for me, it saves us the heartache. If it's got black blotches on the ventral hind wing, we call it Yakima. There's no more of this nigrescens luciform nonsense. We can put that Pelham guy to rest.
Yeah, that you incidentally started, yes.
Yeah.
Just saying.
Just saying.
Awesome.
Okay, well, thank you so much, Caitlin. We are so thrilled to be the first to hear about this in a public forum. I want to remind everybody, please, though, to not post this on social media, not to make it public, because Caitlin and John still have some work to do to get this official, and they want to be the ones to be able to do that. So if everybody could just keep a little bit of a wrap on it, that would be great.
Yeah, we'll be sure to let everybody know once we get it published, and we'll send out the paper and all that. So.
Okay, thank you so much, and thank you for sharing this research, this fabulous research with us tonight, Caitlin.
Thank you.
Okay.
Thank you.
All right. Thanks, everybody.
Good night, everybody.
Good night.
Bye.
Bye.
Come on. I gotta leave. Gotta go. Gotta get away. Gotta go, go, go. Bye-bye. I don't know how to get out of here.
Yeah, I'll end it in just a moment. Gary wants to know about the recording, so I'm just gonna send him a message real quick. So I think it'll be accessible via the WBA website. Carolyn, our wonderful IT person, usually makes it available. So I'll just, there you are, Gary. Okay, so otherwise, contact me or Carolyn.
Boom, boom, boom.
Okay, so just sending you that message. Okay, anything else?
Boom.
Stuart, you were awfully quiet. Do you have anything to say?
Well, I'm not feeling very noisy today, that's all.
Oh.
Nothing to say, really.
Oh,
Well, I
okay.
made up for you, Stuart.
You always do. Yeah. Okay, well, thanks, everybody. I'm gonna go ahead and end the meeting. And thanks again, Caitlin.
Bye.
All right, good night, everybody.